Hello, my name is 정찬영, and the title of my research proposal is exploring the potential of KDS-2010 as a novel pharmacological therapy for addiction, regulating tonic GABA release by ventral tegmental area astrocytes. Addiction is a complex and debilitating condition characterized by intensive craving for a particular substance or behavior, coupled with compulsive engagement in the desired activity, despite negative consequences. Even though addiction is a significant and escalating societal problem, existing therapeutic interventions have several limitations. The addictive substance includes cocaine, nicotine, opioids, alcohol, and etc. To understand addiction, it is important to understand the mesolimbic pathway. This pathway comprises dopaminergic neurons originating from the VTA and projecting to the NAC. When the behavior stimulates the dopamine release in this pathway, the dopamine release reinforces the behavior so that the behavior is motivated, which means that the animal tends to perform the behavior again. Virtually all of the addictive drugs induce dopamine release in the mesolimbic pathway through driver's mechanisms. Inducing dopamine release is a common action of addictive drugs, and this is why those drugs are addictive. The next keyword to introduce is astrocytes. Astrocytes are star-shaped glial cells, the non-neuronal cells in the nervous system. Astrocytes perform many functions, such as supporting the neurons, regulation of neuronal communications, and regulation of ion concentrations in the extracellular space. A recent study by Yang et al. Revealed the role of VTA astrocytes in promoting the dopamine release from the VTA to the NAC in response to cocaine. Let me describe this study more in depth. VTA neurons include NAC-projecting dopaminergic neurons, namely DA-NAC neurons, and the GABA neurons that inhibit DA-NAC neurons. The study showed that VTA astrocytes tonically release the inhibitory neurotransmitter GABA via SWEL1 channels. Then GABA-mediated tonic inhibitions to the neurons occurs by GABA-A receptors expressed on the neurons. In response to cocaine, tonic inhibition to the GABA neurons is increased. Then the inhibited GABA neurons firing is decreased. Because the GABA neurons inhibit DA-NAC neurons, the decrease of the inhibition, so-called disinhibition, caused the DA-NAC neurons firing to increase. This is how VTA astrocyte promotes dopamine release from VTA to NAC. Recent studies emphasize the importance of glial cells in the study of neurological disorders. Comprehensive approaches to studying neurological disorders, considering both neurons and glial cells, are necessary. So our study aims to investigate whether the tonic GABA release from VTA astrocytes also contributes to dopamine release in the case of other addictive drugs than cocaine. Also, our study aims to determine whether inhibiting astrocytic tonic GABA release through the pharmacological agent KDS-2010 can have therapeutic effects on addiction. Our first aim starts with identifying the DA-NAC and GABA neurons. As described in Yang's paper, DA-NAC neurons can be identified by retrobeads injection and TH staining, and identifying the neurons with both green and blue fluorescence. Also, GABA neurons can be identified by using GAD2 and cherry mice. After the identification of the neurons, quantifying tonic GABA current can be accomplished by using the baseline whole cell patch clamp recordings. Tonic GABA current can be measured by the change of baseline induced by BIC, a GABA-A receptor antagonist. Cocaine potentiates tonic inhibition onto VTA GABA neurons. Our hypothesis is that addictive drugs other than cocaine will amplify the tonic inhibition in GABA neurons just like cocaine. After investigating the role of VTA astrocytes in addictive drugs, we will determine the effect of KDS-2010 on addiction as a second aim. MAO-B is an enzyme responsible for astrocytic GABA synthesis, and KDS-2010 is a MAO-B inhibitor. First, we hypothesize that the administration of KDS-2010 will attenuate the increased tonic GABA current in GABA neurons due to addictive drugs. We will also determine the effect of KDS-2010 on addictive behavior. Conditioned place preference, or CPP, is a phenomenon that a mouse prefers to stay more in the chamber in which the mouse got a cocaine injection. We hypothesize that CPP induced by addictive drugs will be reduced by the KDS-2010 administration. Our study may have pitfalls and alternatives. First, other drugs than cocaine may induce dopamine release through mechanisms other than astrocytic tonic GABA release. However, this study holds significant value in the expectation that KDS2010 could show a therapeutic effect on cocaine addiction. Also, KDS2010 may not effectively reduce estrocytic flavor release. In this case, SORA1 channel inhibitor such as DCPIB would be worth testing its effect. The aim of our proposed study is as follows. This study aims to provide novel insights into treating addiction by targeting associated mechanisms involved in addiction. Furthermore, the findings may pave the way for developing more effective therapeutic strategies and contribute to advancing our understanding of the role of astrocytes in addiction-related neurobiology. Thank you for listening.